Hello everyone, it's great to be joining you all today. My name is Brendan Daisley and I'm a PhD student in Dr. Gregor Reid's lab at Western University, located in London, Ontario, Canada. Today I'll be sharing with you some of the exciting findings from my PhD project, which focuses on how beneficial bacteria can be used outside of human health to address big picture problems in the world. For example, in reducing the decline of important insect pollinator species like honeybees. But before I get to that, I just want to first mention how truly honored I am to be receiving the Armand Frappier Outstanding Student Award. Among his other accomplishments, Dr. Armand Frappier was instrumental in the development of a vaccine against tuberculosis, a triumphant legacy that lives on to this day, and is even helping current-day scientists fight the battle against the widespread COVID-19. To begin the discussion today, I'd like to first quickly touch on the global events that have transpired and that we've all experienced over the past several weeks. While the COVID-19 crisis has been truly devastating and saddened many of us through lives lost, the spread of SARS-CoV-2 isn't the first microorganism and won't be the last to cause a global pandemic. Despite vigilant efforts, it remains a challenge to eliminate problematic species with a known propensity for pathogenicity. However, humans are not alone in facing these challenges. Many wildlife species also face a similar crisis, though their struggles are often under-realized and don't gain the same level of attention as something like COVID-19 pandemic has. Importantly, up to 60% of human pathogens are zoonotic, meaning they can infect both humans and animals, and up to 75% of emerging diseases are estimated to be derived from an animal source, as is suspected in the case of COVID-19. Some other good examples that are likely familiar to most of you is the spread of Lyme disease by ticks and the spread of malaria and West Nile by mosquitoes. There are numerous other examples, however the point to gain here is that while humans and animals' health are often viewed as separate entities, in reality they're inevitably interconnected and interdependent on one another. Highlighting this further, the term planetary health has made some big headlines recently, and when put simply, refers to health of human civilization and the state of a natural system on which it depends. More than a definition, however, planetary health is a movement that aims to safeguard human health through extending the diligent health measures we put forth for ourselves towards that of wildlife as well, as a starting point. The end message being that the healthier we can keep our planet and the animal communities within, the better off humans will be as a result. Though infectious disease is not the only problem faced by humans and animals alike. Another important aspect is exposure to environmental toxins, such as pesticides. I'll discuss the specifics later in the talk, though intuitively, it doesn't make much sense that we wear full-body protective gear when applying these chemicals to our crops, yet consume foods that come from these exact same fields without any protection at all. Insects are particularly at risk, as demonstrated by a 41% decline in their total populations over the past decade. While not highest on the list, honeybees were recently declared the most important living thing on Earth, as determined by an international panel of experts at the Earthwatch Institute. And for good reason. Firstly, honeybees are critical pollinators that help support over 1/3 of the global food supply. Through increasing agricultural pollination and subsequent crop yield, they greatly affect food price and availability. Moreover, they are also important to ecosystem health through supporting the reproduction of numerous plant species. The problem, however, is that honeybee populations have been steadily declining on a global scale for the past 30 years, bottoming out around 2007 due to a phenomenon known as colony collapse disorder. The exact cause of this disorder remains elusive, though the general consensus is that infectious disease, pesticide exposure, and habitat loss are the major factors involved. In addition, there is evidence to suggest that transmissible disease in honeybees also threaten bumblebees and other important wild pollinators. Getting back to the fact that many diseases are zoonotic and are transferable between multiple species, including that of humans. And it's not just a problem of one single species. We have to look at everything as a whole. Due to large-scale coordinated response efforts, there has been a slight rebound in honeybee population size since the major collapse in 2007. These efforts include the implementation of disease containment methods, such as burning of the entire apiary at the first sign of infection to prevent subsequent disease spread, as well as petitioning against certain pesticides, such as neonicotinoids, which have been shown to suppress honeybee immunity and increase susceptibility to infection. While these strategies have helped immensely, they have not solved the problem completely, and annual colony losses continue to exceed the limit of sustainability. For example, in recent years, there has been between 25 to 45% total colony loss per year in the US, and similar numbers elsewhere in other countries, whereas the acceptable limit for sustainability is estimated to be between 10 to 20%. Thus, it's approximately double the limit of sustainability. In addition, recent changes in administrative decisions have led to relaxed laws around the usage of many dangerous pesticides like neonicotinoids, which greatly benefit agricultural crop yields, but further burden honeybee populations. So, how do we properly address this situation? The complete removal of pesticides isn't currently an option, nor is the complete avoidance of all infectious disease. Thus, an alternative solution is needed. Some possible solutions that have been tried already include banning of the most toxic pesticides which threaten honeybees, which has been successful in some European countries, but are often replaced with an equally or more toxic alternative. Environmental bioremediation. This does have potential, but is yet to be implemented on a large enough scale to be effective. Selective breeding of bees. This has also been attempted, but has largely failed in producing the desired phenotype, and in some instances, selective breeding can actually lead to the creation of more dangerous bees, such as Africanized killer bees that now pose a human danger in some parts of the world. Organic farming practices. Good as well, but is financially difficult to sustain and drives up food prices. Antibiotics to control pathogens. Good in some situations, but can lead to antibiotic resistance and have deleterious effects on the host as seen in humans. Even the idea of robotic bees as replacement pollinators has been sensationalized on some popular TV shows. To date, though, none of these have been successful. Thus, we believe a simpler approach may be supplementing honeybees with beneficial bacteria, such as probiotic lactobacilli. While this idea may seem crazy at first, there is actually a surprising amount of rationale to support it. For example, lactobacillus species are well-known symbionts in honeybees that have the capacity to act as major modulators of health. Also, due to undergoing reductive evolution, the honeybee genome is genetically depauperate in a number of key immune and detoxification genes. Thus, honeybees rely heavily on microbial constituents for proper immune function and metabolic competency required for complete detoxification of pesticides and other environmental chemicals. To investigate this further, we tested the theory in our Drosophila model, which we used as a predictive high-throughput screening platform to identify strains of lactobacilli with an ability to improve insect immunity and detoxification. This work largely was performed early on in my PhD thesis and helped to elucidate several strains of lactobacilli with promising probiotic potential, ranging from the ability to decrease toxicity of organophosphate insecticides to mitigating pathogen susceptibility to improving immune function during neonicotinoid exposure. Thus, the beneficial effects associated with supplementing these probiotic lactobacilli overlapped near perfectly with two major stressors deemed responsible for high honeybee colony loss: infectious disease and pesticide exposure. A question you may be wondering at this point is, how exactly can one supplement honeybees with probiotics? So the task now was to develop a delivery method to enable testing of these strains in honeybees. But first, we have to consider the complex anatomy of the hive. To give you a brief overview, here's a short video summarizing the life cycle of bees in the hive.
Here is a bee egg as it hatches into a larva. Those newly hatched larvae swim around their cells, feeding on a liquid food that nurse bees secrete for them. Then their head and their legs start to differentiate as they transform into pupae. Here's that same pupation process seen from above with varroa mites running around in the cells. Next, the tissue reorganizes in their body and the pigment slowly develops in their eyes. In the last step, their skin shrivels up and they sprout hair.
So, the idea is that we want to supply probiotics to the adult bees, but also the young larvae, which you just saw in the video, which are at the highest risk of infection due to their weaker immune systems. To do this, we must be able to disperse the probiotic bacteria through the brood chamber, where both honeybee larvae that are in their early developmental stages are, as well as the adults that help feed them, that are referred to as the nurse bees. There are three basic delivery methods that one could use to attempt to deliver probiotic bacteria to the hive. The first and simplest is a freeze-dried powder-based supplement. The benefits being that it is easy to administer, though the disadvantage is that it doesn't facilitate active oral consumption by the adult nurse bees. The second option is delivery through a sucrose syrup, which is often supplied to bees by beekeepers as a source of quick energy. Though this method would facilitate good oral consumption of the probiotic strains, it unfortunately also kills the bacteria due to osmotic stress. A recent study evaluating this found that even at the conservative temperature of 30 degrees Celsius, which is far lower than what would be expected in the hive, demonstrated a near complete reduction in bacterial cell viability after only 96 hours. The last option is delivery through something called a pollen patty, which is essentially a nutrient supplement for bees. The benefits are 1. Good oral consumption, 2. Bacterial cells remain viable for well over 2 weeks, and 3. This method also provides essential nutrients to the hive. Thus, for these reasons, we chose to use the pollen patty for our delivery method of choice. To give you a brief background, pollen patties are a type of bee supplement common to the industry. They provide essential nutrients and are frequently used in apiaries to ensure nutritional adequacy of honeybees. And because pollen patties are often made on site by beekeepers themselves, the idea is that probiotic lactobacilli could be easily incorporated during this process, thereby making this delivery method easily adaptable in practice. Thus, to test this theory, we designed a probiotic-infused functional food item using the pollen patty as a backbone, which we termed the biopatty. Making the design as realistic as possible, the base of the biopatty contains the same 3 nutrient sources which make up the standard ingredients that are already in use in commercial patties. In addition, however, the biopatty contains 3 strains of lactobacilli based on our previous studies in Drosophila, each with a unique function and a purpose in mind. The first is a Lactobacillus rhamnosus strain, added for purposes of pesticide detoxification. The second is a Lactobacillus plantarum strain, added for purposes of immune regulation. And lastly is a Lactobacillus kuhnkeii strain, originally derived from a healthy honeybee for the purposes of microbiota restoration and digestion. To give you a glimpse of how we make these, here is some photos of what making the biopatty in the lab looks like. In the bottom left is Christina Torres, a student from our collaborators lab, mixing the biopatty, and in the top right, the biopatty being poured onto a sheet of wax paper, prior to being sandwiched by another sheet before being put in the hive. And here is a schematic of how the biopatty is supplemented to the hive, and a photo of what one actually looks like inside a real hive. The idea is that by putting the patty on top of the brood chamber frames allows both adult bees and young larvae to gain access to the probiotic strains via communal dissemination of food sources in this area of the hive. Although my thesis and research have applications beyond honeybees, the focus today will only be on bees, and thus the hypothesis is specific to these insects, and is that hive administration of Lactobacillus species can enhance honeybee resistance to pesticides and pathogens. I've discussed with you already some of the work that has been done to identify probiotic lactobacilli with the capacity to improve insect immune function and detoxification, and some of the work that has went into developing a delivery method for these lactobacilli. The third aim of my thesis is to test the identified probiotics against pathogens in real bees under natural conditions. And the fourth is to test the probiotics if probiotics can reduce pesticide toxicity in bees under natural conditions. However, for the purposes of today's talk, I'll only be discussing the latest findings relevant to aim 3. Now, on to some of our exciting findings thus far. The idea was simple: supplement hives with the probiotic biopatty and then monitor disease prevalence. Briefly, our experimental setup consisted of a control group that received no treatment, a vehicle patty group which received the base patty ingredients, and a biopatty group which received the base patty ingredients alongside the 3 probiotic candidate strains identified in past experiments. Both a no treatment control group and a vehicle control group were included to ensure that if any differences we saw in the biopatty group were indeed from the added probiotic strains and not from the added nutrients in the background patty itself. Subsequently, we would measure brood count in the hive as a proxy of overall health, determine palatability of the biopatty to the bees, and also sample honeybees at different developmental stages for in-lab assays in which we would dissect them, measure immune parameters, and quantify pathogens of interest using culture-based and/or molecular-based methods. However, after an unfortunate turn of events, and only after 12 days into our first field study, a qualitative in-field assay known as the rope test indicated signs of an American foul brood outbreak, a deadly honeybee disease known to be caused by the bacterial pathogen Pentabacillus larvae. We then confirmed these results in lab via microscopy, 16S, ribosomal RNA gene sequencing, and ERIC typing, all of which were consistent in identifying the suspected pathogen. Following this, we contacted the Middlesex bee inspector, who swiftly met us on site the following day. After performing a number of qualitative tests, mostly relying on visual and/or olfactory-based cues, the inspector determined that an entire apiary needed to be shut down immediately to prevent the spread of disease to nearby apiaries. Following Ontario law, we took our last samples, closed off the hives, euthanized the bees, and then torch sterilized the hive box frames. Here, you can see the extreme measures deemed necessary for preventing spread of AFB through flame sterilization. Though a truly unfortunate situation, it was eye-opening to see just how rapidly an entire apiary could be wiped out, and to think about the repercussions this might have on someone who is relying on their bees as a primary source of financial income. Interestingly, despite P. larvae spores being nearly ubiquitously found in honeybee hives worldwide, disease symptoms only arise when P. larvae transitions to its active growth stage and reaches high levels in the hive. This process is not fully understood, but is suspected to occur when the honeybee immune system becomes weakened due to natural causes or from exposure to immune-suppressing pesticides. Once this happens, however, the pathogenic bacteria causes disease by infecting young honeybees, burrowing through their gut wall defenses, and then feeding on them from the inside out. The only defense that beekeepers have is to prophylactically use antibiotics to suppress this pathogen at key times throughout the season. After all was said and done, the positive side of things was that we had collected an ample number of samples throughout the experiment, which allowed us to go back and analyze how each of the different treatment groups impacted infection dynamics. We did this by homogenizing the collected larval samples, spot plating the homogen on selective agar, incubating the samples to allow growth of the P. larvae, and then enumerated pathogen load associated with the larvae from each of the treatment groups. Our findings were exciting, and showed that despite no differences in pathogen load at the start of the experiment, honeybee larvae from the biopatty treated hives demonstrated significantly lower levels of the pathogen P. larvae at day 12. In addition to measuring pathogen loads, we also performed a colorimetric-based protein hydrolysis assay to measure pathogen activity. Interestingly, while similar trends were observed in the biopatty treated group showing the lowest proteolytic activity out of all groups, the vehicle patty group demonstrated significantly higher pathogen activity than both the no treatment control group and the biopatty treated groups. This suggested to us that while essential nutrients may be beneficial under healthy conditions, they could potentially exacerbate infection during active outbreaks of AFB. Past studies are limited, however, do support this notion, as has been demonstrated with commercial pollen patties increasing the proliferation of honeybee fungal pathogens during experimental infection. Nevertheless, while future studies are necessary to confirm this, the added probiotic lactobacilli in our case appeared to largely mitigate these effects, suggesting a potential role for pathogen exclusion during supplemental feeding, which is commonly practiced in apiculture. Going forward, to deconvolute the complexity of the hive, we set out to confirm our findings from the field trial by performing controlled P. larvae infection assays on laboratory-reared honeybee larvae. Briefly, we did this by collecting first instar larvae from the hive, which had been laid within the past 24 hours by the queen. We then acclimated them to the lab conditions with or without the prophylactic supplementation of 3 probiotic strains used in the biopatty, and then infected them with standardized dose of P. larvae spores. Subsequently, we measured survival, pathogen load, and assessed transcriptional changes to immune-related genes. Validating the assay, we showed that the uninfected PBS control and probiotic control groups both had similar levels of survival during the monitoring period, nearing 90%, and was expected. Next, we found that the P. larvae infected group that was pre-treated with the PBS vehicle had significantly reduced survival, with over 30% of individuals succumbing to infection by day 5. Conversely, groups that were infected but received pre-treatment with the 3 strains of lactobacilli experienced significant improvement in survival, which was comparable to that of the uninfected control groups. Further corroborating our field study findings, pre-treatment with the 3 strains of lactobacilli that were present in the biopatty also significantly reduced the number of P. larvae cells found inside of infected individuals compared to those that received the PBS pre-treatment. Thus, together, these findings suggested that the pre-treatment with probiotic lactobacilli strains may be improving pathogen control through upregulating the host innate immune response in bees. In screening several immune-related genes, we identified that the antimicrobial peptide, defensin-1, was consistently upregulated by lactobacilli under both infectious and non-infectious conditions. Importantly, defensin-1 is a critical antimicrobial peptide produced via activation of the IMD pathway in honeybees, which is in a highly conserved innate immune response in insects. And also, when isolated, defensin-1 has shown to elicit strong antibacterial properties against P. larvae. These findings are consistent with our previous work using a Drosophila model, in which we showed that oral supplementation with LP39 could robustly increase the expression of diptericin, which is an analogous antimicrobial peptide to defensin-1 that is produced in response to IMD pathway activation. The proposed mechanism for this response is the unique type of peptidoglycan present in LP39 cell wall. Specifically, LP39 produces DAP-type peptidoglycan, which is unique as most other lactobacilli produce lysine-type peptidoglycan. The importance of this is that DAP-type peptidoglycan, when purified and in isolation, has been shown to be capable of directly activating the IMB pathway and can upregulate a broad range of AMPs. Of relevance, a recent study screened the large majority of lactobacillus species naturally found in honeybees and found that none of them possessed DAP-type peptidoglycan. Thus, this may also explain the multiple failed attempts at using honeybee-derived lactobacillus species to improve resistance towards American foul brood disease caused by Pentabacillus larvae. Importantly, these findings, together with our own, help in narrowing down the mechanism and suggest that pretreatment with the LP39 containing biopatty acts to improve pathogen resistance against P. larvae through providing unique beneficial immune stimulation that the native microbiota is incapable of. So, quickly recapping so far. At this point, we now know that the 3 strains of lactobacilli we tested could reduce pathogen load in the hive and improve survival against deadly infection under controlled conditions in the lab. Which begs the question of whether the biopathic treated hives in the original field trial actually required burning, or whether they might have been able to recover if given time. Our future work aims to investigate this in an isolated location where we can monitor the infected hives without the risk to nearby apiaries. However, we also wanted to see if these same 3 strains of probiotic lactobacilli could be used to address the overuse of antibiotics in apiculture, which is a substantial contributor to the spread of environmental antibiotic resistance. Oxytetracycline is one of the most commonly used antibiotics, often as a preventative measure against Pentabacillus larvae to halt American foul brood outbreaks from occurring, but is also frequently used in other sectors of veterinary medicine as well. An important aspect of oxytetracycline is that it's a bacteriostatic antibiotic, which prevents growth of the target pathogen and allows honeybees to clear it from their systems, but is unable to kill the pathogen itself. It is often used in a cyclic manner to prevent pathogen loads from getting too high in the hive, but poses a serious risk to honeybees through its deleterious effects on host nutrition, immunity, and the honeybee gut microbiota. Thus, the experimental setup was similar to the first set of field trials. In this case, however, a 2-week standard course of oxytetracycline was given prior to further treatment, which once again consisted of the same treatment groups as our first set of experiments containing a no treatment control, a vehicle patty group, and a biopatty group. Here's a quick look of how we collected the larvae using sterile forceps and adult bees using sterile 50 ml Falcon tubes. So, similar to humans, honeybees possess a core stabilized microbiota structure that confers a variety of health benefits to the host. Our results demonstrated that standard oxytetracycline treatment could significantly lower total levels of these important core species. Moreover, the abundance of several of these key symbionts, which are known to regulate immune function and nutrient metabolism, such as Frashella pereira and Lactobacillus firm 5 strains, were significantly depleted in response to oxytetracycline treatment. In addition, the standard course of oxytetracycline also significantly increased total abundance of TEP-B, an efflux pump resistance gene in the honeybee gut microbiota. Further investigation showed that this was largely due to the oxytetracycline-induced increase in relative abundance of the gram-negative gut species Gilliamella apicola, which correlated strongly with TEP-B abundance. This is important as environmental spread of antibiotic resistance threatens humans' health through increasing multi-drug resistant pathogens that plague hospitals, and can also increase bacterial virulence of pathogens in wildlife species exposed to subtherapeutic levels of antibiotics. Investigating how these changes in the honeybee microbiota may affect overall colony dynamics, we enumerated brood count in the hive frames using a semi-automated counting method, which is ultimately a good proxy of individual nutrition and overall hive performance. The findings demonstrated that exposure to oxytetracycline significantly reduced hive performance after 2 weeks of treatment. Alongside past studies also showing tetracyclines to perturb the gut microbiota and increase honeybee mortality, these results shed light on how reduction of key nutrient-promoting symbionts, like lactobacillus species, leads to a detrimental effect at the colony level. Past comparative studies have also shown that thriving colonies with high productivity exhibit a very distinct microbiota structure that is dominated by a small number of well-established chorophyll-type members, with very low levels of other species present. Intriguingly, however, the biopatty supplementation showed a strong ability to rescue hive productivity following 3 weeks post-exposure to the antibiotic, with benefits extended all the way until the 4th week of supplementation. Thus, we wondered if the positive effects of the biopatty supplementation may be elicited through improved recovery of the microbiota following antibiotic exposure. To test this, we generated Bland-Altman-like effect plots to compare the endpoint microbiota structural differences between biopatty supplemented versus vehicle supplemented individuals. What you're seeing here is the plotted difference in relative abundance of each SV between groups, delta A, plotted against the variance, or within group difference in relative abundance for each SV, delta W. Accordingly, this acts to clearly highlight the dominant bacterial species, or rather core microbiota members, which are found in nearly all of the samples at low variance on the left. As well, the SV points on the right represent rarer species which show high inter-individual variance, most of which are transient opportunists that honeybees likely come into exposure with during foraging. Interestingly, we see a trend of nearly every one of the core microbiota members being increased in the biopatty supplemented group, yet none individually reached significance. To investigate the underlying compositional changes responsible for these observations, a nested subgroup analysis was performed and showed that, while there is no change in the relative abundance of these core cluster SVs, there was a significant reduction of rarer SVs in the biopatty treatment group. These findings were supported by several other indices of microbiota diversity, including reduced beta diversity, meaning that individuals had a more similar or stabilized microbiota within the biopatty supplemented group, decreased species richness, and a trend towards increased species dominance. Thus, together, these results suggest that the probiotic strains in lactobacilli may help to support microbiota recovery following antibiotic exposure by suppressing the growth of unwanted opportunistic species, as opposed to directly stimulating the growth of core microbiota members themselves. Next, and most importantly, we assess the efficacy of oxytetracycline in reducing P. larvae as a prophylactic treatment, as it is purported to do. First, we measured the effect in larvae, and found that after 1 week of standard treatment and onward, there was a significant reduction in pathogen loads. However, in larvae, which can act as large reservoirs of the pathogen in the hive, oxytetracycline was ineffective at lowering pathogen load, which suggests the increased tetracycline resistance from core symbionts in the gut, as shown earlier, may be spilling over and providing P. larvae safe harborage from antibiotic exposure, which is a novel finding. Following antibiotics, biopatty supplementation was shown to suppress P. larvae levels lower than that of the controls, and significantly beyond that of antibiotic treatment alone. Interestingly, we also found that biopatty treatment could decrease P. larvae loads in adult honeybees as well, thereby offering a distinct advantage over antibiotic treatment by itself. Thus, this presents a potential opportunity for probiotic lactobacilli to be used as an adjunct alongside current antibiotic therapies. The idea being that reducing pathogen loads more extensively in the hive will lead to a longer period of cessation between necessary treatments, ultimately leading to a reduction in antibiotic usage as well, and thereby reducing the environmental spread of antibiotic resistance. Given that the biopatty demonstrated a bactericidal effect against P. larvae, even in adult bees, we asked the question, how exactly may the probiotic lactobacilli be killing P. larvae? We showed that co-culture with the 3 strains of lactobacilli, or their supernatant alone, were able to significantly inhibit the growth of P. larvae. We also found that 2 out of 3 of the strains in the biopatty were equally as effective as oxytetracycline at inhibiting P. larvae, though it was still unclear if P. larvae were being killed or if their growth was just being inhibited during these in vitro assays. To address this, we co-incubated one of the lactobacilli strains, LP39, with P. larvae for 1 hour and then stained the cells using Cytox green, a selective fluorescent dye that stains only dead cells based on cell wall permeability. Single cultures of live P. larvae and LP39 showed very little green uptake, indicating a high level of cell viability. Upon exposure to 70% ethanol, which was used as a positive control, both single cultures demonstrated a significant uptake of Cytox green, indicating cell death. Interestingly, upon co-incubation, there was a significant uptake of the selective fluorescent marker in only the P. larvae cells, which are differentiated based on their elongated cell morphology, where the shorter LP39 rods showed minimal Cytox green uptake. Together, this suggests that LP39 may secrete an uncharacterized metabolite able to induce cell death in P. larvae, and represents a secondary mechanism by which probiotic lactobacilli can inhibit pathogen load in honeybees, though further work needs to be done to clarify and identify this component. So, to conclude in today's talk, delivering probiotic bacteria to honeybees using the biopatty was found to be safe and effective at reducing the prevalence of a major honeybee pathogen responsible for high levels of colony loss. Of the 3 strains of lactobacilli we tested, some demonstrated the dual capacity to upregulate innate immunity and directly inhibit P. larvae in both early developmental stages as well as later developmental stages of adulthood, which offers distinct advantage over current disease control methods that are being used in apiculture. And lastly, biopatty supplementation was able to boost the beneficial effects of standard antibiotic treatment while mitigating some of the negative side effects it had on the microbiota as well as hive productivity, suggesting it could be used as an effective adjunct in beekeeping to improve honeybee health and reduce colony loss. During the remainder of my PhD, I aim to address the effects of probiotics on other pathogens, as well as test it against pesticide toxicity in honeybees. Future studies, which have already begun, focus on improving the health of honeybees in California, where 1.5 million hives are imported for the purposes of almond crop production each year. Unfortunately, however, almost 80,000 of these hives die each year due to infectious diseases and exposure to toxins like pesticides. This amounts to an astonishing financial burden to farmers, beekeepers, and the end consumer. If we can improve the health of these bees and reduce even a fraction of these hive losses, it could make a substantial difference globally. Here's some photos of the team in California during our setup last spring, our network of collaborators in Los Angeles, as well as some local bee art just outside of the campus in Davis, California, really showing just how important bees are to the economy and local community in this region. In addition to testing in California, we have ongoing field trials in rooftop bees located in Detroit City and in several locations here across southwestern Ontario. If you or any beekeepers you know might be interested in getting involved, please share my contact details with them. With that, I'd like to thank my supervisor, Dr. Reed, for the immense opportunities he's provided me during my graduate school experience. I couldn't imagine being in another lab, and I've had truly an amazing time joining him on these adventures over the past few years during my PhD. I'd also like to thank all of the collaborators who have helped out along the way, specifically bee expert Dr. Graham Thompson, as well as Andrew Pitek, who have helped share a wealth of knowledge about the fascinating world of bees. In addition, I'd like to thank Mark Trinder for his excellent work he did in early pioneering of our Drosophila model for application and predictive usage with relevance to honeybees. And finally, a big thank you to all scholarships and funding agencies that have helped foster travel experiences, networking opportunities, and that have made our global collaborations possible. With that, I thank you all for your attention, and I welcome any questions you may have. Thank you.